Hello everyone, welcome to the video tutorial on how we are going to construct a phylogenetic tree using MEGA11 software. So if you download it from your browser and install it in your system, this will be your interface for MEGA11, where MEGA stands for Molecular Evolutionary Genetic Analysis. So tell me, have you ever wondered how scientists trace evolutionary data and set the evolutionary relationships between species and sequences? We have always heard about, okay, these were our ancestors from the amoeba to apes to human, and the all phylogenetic tree we have learned. How do they construct this? How do they collect these data? So you are going to be part of this by using this simple tool, and let's get some sequence, for example, and construct our own phylogenetic tree and learn the evolutionary relationships in species a little better. So just get in NCBI. So you can use any nucleotide sequence, or you can use the protein sequence for this. So for example, we are taking the nucleotide sequence here, and let's say human cytochrome C. Okay, we can take this for example, cytochrome C genes from Homo sapiens, and so here we got our cytochrome C gene from Homo sapien, and you can select any one of this. Let's just say we can select this as it's a small sequence as compared to other, or we can also select this. That's a partial series sequence of Homo sapiens. As the GenBank data you get here, and I have explained all these details we get from the NCBI, and you can just check on that, or you can just click on Run Blast. You can also directly go to Blast NCBI, and then you select your gene and save it, and you will have to select one of these in that also, the nucleotide, the protein, the BLASTX, and the translational nucleotide sequence, whatever you are selecting for your experiment or you want to do it to collect the data. You can also select the file from here if you want, and just go here and blast. So you will get this blast data in here in few minutes. So here we got our blast data of the nucleotide sequence we selected, and you can just scroll down here and see the RID, the program blast, and the database, the query ID, the description of our gene, the molecule type, that is nucleotide, and the query length, that is 956. You can also add any organisms here if you want the same sequence in different organisms, or you can just see the result here. So let's say these are the 100 sequence selected in one run. So we will just select few of them and take them for the example to construct our phylogenetic tree. So let's say you can select any 10 or 5 from here, but what we will do is we will get one sequence from different organisms. Let's say we are going to select this cytochrome C somatic mRNA gene from Homo sapiens and scroll down. Let's say we'll get one sequence from troglodytes here, You can select any of them. One sequence from Paniscus, one sequence from Gorilla. Other, you can select the Hylobates, the Pongo abeli, the Symphalangus. So I guess these are enough for example. So we have selected the 7 sequence and it's okay. We have selected them and you just need to click on download here. We will go with a FASTA aligned sequence because alignment is important. So it will be here in the downloaded section. And let's just go back to MEGA11. So in MEGA11, you just need to click first on the align and edit and build alignment. So this alignment editor will be opened. So you're going to select create a new alignment and then click on OK. Here they will ask you're building a DNA or protein sequence alignment. So we will just select DNA for this. This is here our alignment. Okay, so now what we need to do is click on edit and insert the sequence from file. So this file will be the downloaded file, this here. Okay, so this will be the result and these are the sequences we have selected and downloaded from BLAST. So what you need to do is you can just click on that and you need to select these sequences and align these sequences. So again here select sequences and we are selecting all that we have selected. So that's how we have selected and you are going to I will just drag it up here. Okay, so we are going to click on alignment. We are going to align it using cluster W. That's an algorithm. These are the open gap penalty and gap extension. These are part of the this cluster W method. If you want to understand these metrics and algorithm, you can comment and I will explain it to you in another video. So in this you are going to keep it the way it is and click on OK. And alignment is taking place, pairwise alignment, multiple sequence alignment, and here we got the aligned sequence here. So these are the aligned sequences and you can just save now. Here data, you can click on data and click on export alignment in mega format because we are going to use this file for constructing phylogenetic tree. You can give it any name and you can just phyllo tree. For example, I'm giving this a name on desktop. And again, that's how we are going to save our data, protein coding nucleotide sequence data, because these nucleotide sequence code for proteins. And now you can minimize it. Here you can see a file here. So now just click on phylogeny and we are going to construct a tree using neighbor joining method. There are different methods like maximum likelihood, the UPGMA and maximum parsimony, and different algorithms that are used to construct the phylogenetic tree. But we are going to use neighbor joining method for this tutorial. It is a very simple and reliable tool you can use. So you just need to select the file that you have saved earlier from here and just open it, and here the data will be like that. Okay, so what you need to do is you can select the test of phylogeny. It could be bootstrap method or interior branch test. And bootstrap method is important because it gave you the surety, like if uh, it give you the result by comparing align the sequence about 1,000 times before it show it to you, before it give you the result. You can change the bootstrap value, replication value here. If you can just click on 500 if you want to be it to replicate for 500 times, but let's just say at least you should always go with 1,000 times. So for that you need to do one thing, that you need to select this file here and select only the sequences. As you can see, there are name and sequence like that, it is also selected, but you just need to select the sequence you are interested in and then click on neighbor joining method, and using the current data, the bootstrap value, and just click on OK. And this is in progress, and we are constructing a phylogenetic tree. And here we go. This is our phylogenetic tree. As I have constructed the tree, you can see the things you can edit in here. You can just double click and change the name, like you can just let it be the pen troglodytes here, and you can see likewise you can edit any of those, the length of your tree from layout here. You can use different layout, tree width, the height, this is fine. There will be different tree which would be rooted and which have the outgroup. We cannot use this in that phylogenetic tree, and you can use the font in here, the automatic above the branch, below the branch, or wherever you want them to be, and hide if they are shorter than 0. This value. That's how you can hide or show the length of your branch. These branch length are a detail of evolutionary time, and you can also edit the statistical information here. Like you have the data, you can make it more beautiful. You can uh, have the other display options like in here, the tree cutoff options, the cutoff and condensed value, and you can compute, like you can see in here. This is rectangular now, it could be straight like that. It isn't looking good. You can go with circle one, so you won't be able to see it like this. So you can edit these informations. The tree will be the same, but you can change the layout of or the display of your tree. You can see the bootstrap consensus tree here, and let's just change it to traditional one. Midpoint. The curve also look beautiful like this. So you can now just extract your file, save your file in here like this in a mega format, or if you are going to use this in your research paper, you can just copy the image like this and paste it wherever you want, or you can export the current session the tree, the group name, any data you want. And you can also save the file in PDF format, in PNG format, anyhow you want. You can, let's say PNG file name is Philo. Let me bring it here. Philo tree. Our tree has been saved. We can just see in here. Here we go with our phylogenetic tree. This will be like this, and you can edit it as you want. So I hope you understand how you can use the MEGA11 software to construct a phylogenetic tree. Thank you for watching. Please like and share.